Our bodies are made of cells, trillions of them. There are 5,000 times more cells in our bodies than there are people on the planet. The range of cells in the human body is extraordinary, from neurons that are over 1 metre long to immune cells trained to seek and destroy invaders, and cells that literally build your bones.
For centuries, we scientists have been studying cells. Our aim? To uncover the deep secrets of how they work, how they make our bodies work, and crucially, what happens when they cause disease.
In the past, we would study a big group of cells at once and come to one conclusion for all of them. In cancer, for instance, we'd look at a tumor sample from a biopsy, which contains many types of cells. We'd learn a lot, but it would be an average. It wouldn't tell us anything about the variety of cells or how each cell contributes to the disease.
Now we are turning the study of cells on its head. We're using a technique with a complex name but an elegant concept: cellular transcriptomics. With this, for instance, we can take a tumour and separate it into thousands of unique cells and then read information from each cell individually. This has never before been possible on this scale.
We've all heard of DNA, but in cellular transcriptomics, what we're interested in is RNA. Think of RNA as DNA in action. It's the set of molecules copied from the DNA master plan that tells the cell how to behave and what to be. The cell uses a remarkable biological machine called RNA polymerase to transcribe DNA and make precise RNA copies. Here we're seeing just one RNA being made, but there are about 10,000 different RNA molecules in each and every cell.
What's amazing is that each cell has a unique pattern of RNA copies. This is a detailed genetic signature, which tells us a lot about its origin and function.
Because we're studying thousands of RNA molecules per cell, we have access to an extraordinary amount of information about the cells in a tissue or a tumour. Far more information than we've ever had before. Where once we were making, say, 10 observations about a cell, now we're making thousands. That means we can understand much more about how cells function and how they differ from each other.
So what can we do with all that information? We're trying to understand how diseases like cancer and immune disorders happen at the level of the individual cell. Many diseases start in the cell. When it malfunctions, disease develops. Now, we can find those cells.
I'm studying cancer cells that lie dormant inside bones, but can wake and cause cancer to relapse.
I work on autoimmune diseases. We believe these start with just one or a few cells in the immune system going rogue.
I'm studying breast cancer, and I'm seeking out the cells inside breast tumors that can stop immune therapies from targeting cancer effectively.
With cellular transcriptomics, we have a realistic chance for the first time of finding these rare disease-causing cells, and we can examine at very high resolution how they differ from healthy cells.
Importantly, we can make use of those differences and seek ways to target disease cells while leaving healthy cells alone.
Cellular transcriptomics has only recently become a reality, but it's already offering an incredible insight into how cells work in health and disease. We can't wait to push the boundaries of cellular transcriptomics even further and to make a difference to our understanding of disease and ultimately the treatment of disease.